To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.
Hello my dear students, welcome to today's lesson. I'm going to continue with the extra questions related to inheritance. So we have been discussing so many questions. Now this is extra question 21. The first part, what are gene mutations? Now you know what genes are. That is a particular base pair sequence that is present in the DNA molecule of a chromosome responsible for a particular character. So if it is a chromosome that is responsible for the production of hemoglobin, we have discussed that under thalassemia. So that has a particular DNA that is consisting of a particular sequence of base pairs. Now this base pair, if it changes, then the character that is produced by that particular gene will also be different. So that is what we call as gene mutation. A change in the DNA molecule of a gene that can lead to a change in the character. That is what we call as a gene mutation. So when the dominant gene that is responsible for the production of hemoglobin gets mutated, then the production of hemoglobin reduces. So that is a disease that is caused by the mutated gene. So I'm sure you're familiar with that concept. If we are to answer what gene mutations are. What are gene mutations? The change in DNA molecule of a chromosome that causes a change in character. That is mutation. Mutation is known as gene mutation. So mainly the change in DNA molecule of a chromosome that causes a change in character is known as gene mutation. Normally when you're answering a question, you don't use the same term given as the question when you try to explain. That's why I have written this mutation within brackets. So it is the change in DNA molecule, because the part of the DNA molecule with a certain base sequence is what we call as a gene that is responsible for a certain character. So when that changes, the character also changes. That is what we call as gene mutation. Then here you can see, write 3 disorders that occur due to mutated genes. So you're familiar with some of the disorders. What are they? Albinism. Albinism is a disorder where the production of melanin pigment is reduced. The gene responsible for the production of melanin gets mutated. Because of that, the melanin pigment is not produced. So because of that, you see an abnormally white color of the skin, the hair, eyebrows, eyelashes, everything is white in color, an abnormal white color because there is no melanin pigment. So that is caused by a mutated gene, albinism. Then you are familiar with thalassemia. Again, thalassemia, the dominant gene is responsible for the production of hemoglobin, but the mutated gene, that is the recessive gene, causes the disease where the production of hemoglobin is less. So albinism, thalassemia. In addition to that, we have discussed some syndromes. You can remember the Turner syndrome, where there is less number of chromosomes. There is only one X chromosome. Normally for a person to be a female, it has to be an XX combination. If the person is a male, there will be XY combination. But if there is only X chromosome, then that condition is known as the Turner's syndrome. Then Klinefelter's syndrome, where you have an additional chromosome. That is also a mutation, XXY. So that is Klinefelter syndrome. Then Down syndrome. That is also a mutation to the autosomal chromosome. So those are all diseases or disorders that are caused by mutated genes. You can write any 3 disorders. So I will say albinism, then thalassemia, and Down's syndrome. Down's syndrome. Albinism, thalassemia, and Down syndrome. In addition to that, you can also write Turner syndrome and Klinefelter's syndrome. So those are all caused by gene mutations or mutated genes. The changes in DNA molecule of a chromosome that causes a change in character is known as gene mutation, and these are examples of disorders that occur due to gene mutation. So I'm sure you all also have written a correct answer. With that, students, we'll move on to the next slide. Extra question 22. Show the occurrence of children between a healthy father and a diseased mother with thalassemia using a suitable diagram. So we discussed that thalassemia is a disease caused by gene mutation. So here it is a healthy father and a diseased mother. Now this, of course, you all have to remember, students, this is caused by gene mutation. So because of that, it doesn't matter whether the father or the mother has the disease, it's going to be similar outcome. Unlike the gene linkage. This is not gene linkage. So it's the same, irrespective of whether the father or the mother has the disease, you will get the same type of children. So here, if you are to show the occurrence, dominant gene is given as capital T and recessive gene is simple t. So you all know capital T, healthy person. Heterozygous condition, capital T, simple t, a carrier person, and simple t. Homozygous recessive condition is the disease condition. So we have a healthy father and a mother who has the disease. So there, if we write the parents, healthy father, capital T, and the mother with the disease, simple t. So if you get the gametes, it's only one type of gamete because both homozygous. So here capital T, this is simple t. And when they combine, the children all are going to be heterozygous. Capital T, simple t. That means all the children are going to be carriers. All are carriers. So you have a healthy father, mother with the disease thalassemia, and all the children are going to be carriers. They can be sons, daughters, doesn't matter. All of them will be carriers. Is that clear to you all? So here, parents, the gene combination is given, or the genotype, capital T and simple t. The gametes are capital T and simple t only. So the children, they are all carriers. Is that clear to you all? So then I'll move on to the next question. Extra question 23. Mutations can occur due to changes in the number of chromosomes. write 2 factors that cause gene mutations. What are the factors that can cause gene mutations? Now, there are gene mutations that are caused by radiation. You're familiar with that. The UV radiation that is there in the sunlight. You're familiar with electromagnetic waves and the UV radiation. This UV radiation can lead to gene mutation. If you're exposed to heavy UV radiation, the genes can get mutated. Then in the hospitals, you all would have seen now in some places where they have this radiology department where they take X-rays, gamma rays for different purposes, for treatment or photographic purposes to diagnose the diseases. They have all these different techniques where they use radiations, high energy radiations. Now these radiations also can cause mutations. So you have to be careful. Then there are some chemicals, chemical substances that are added to food, substances that we use in our day-to-day life. Those also can cause gene mutations. Now, for example, cosmetics, the hair dye, the hair coloring, that has chemical substances that can lead to gene mutations. So, like that, there are other chemical substances also. So, these are ways or factors that cause gene mutations. So, I will commonly say radiation and chemical substances. So, here we can say radiation, or just for you all to know the examples under radiations, ultraviolet rays, then gamma rays, X-rays. These can lead to gene mutation or they can cause gene mutation. Then there are chemical substances. So, hair dye also can cause gene mutation. So, these are things that you need to remember, students. So, factors that cause gene mutation. With that, I will move on to the next question. Extra question 24. Write 3 applications of knowledge of inheritance to improve quality of animal or plant products. So, you all know naturally there are plants and animals that are useful to man. We get milk from cows, we get eggs from hens, then we use the meat of animals like chicken, mutton, those are all meat obtained from animals. In addition to that, we get lot of producers from plants. Rice, wheat, fruits, vegetables, all those are obtained from plants. Now, if we use the knowledge of inheritance and if we can improve the quality of these animals and plants, then that will be more useful to us. So, can you think of the examples? Now, we can have a cow that can produce more milk so that we can satisfy the need of the growing population. So, for that, we can use the knowledge of inheritance. Genetically modified cow. So, who produce more milk. So, you are familiar with gene technology or recombinant DNA technology. So, that is one. Then we can have hens that lay a larger number of eggs, more eggs, and even larger eggs with more nutritious value. That is also possible. We can have pest-resistant crops, weedicide-resistant crops that give more yield. Then also we can produce fruits that are more fleshy, more tastier, more nutritive, and also more number of fruits. So those are all applications that are obtained using the knowledge of inheritance. So you are supposed to write only 3 applications. So I'll write down 3 applications. can produce cows that can produce larger volume of milk. Then hens that lay more number of eggs can be produced. Right. Plants that are resistant to pests can be obtained. So these are three uses. Can produce cows that can produce larger volume of milk. Then hens that lay more number of eggs can be produced. Plants that are resistant to pests also can be obtained. Like this, you can write any of the three uses or applications of knowledge of inheritance. So with that, students, I'll move on to the next question. Extra question 25. The first one, what is recombinant DNA technology? What is recombinant DNA technology? Now, there are different sources that have DNA molecules. Say a DNA from a man, a DNA from a bacteria, or DNA from a cow and a DNA from a bacteria. These are DNA molecules of different sources. If you combine them together, take part of one DNA and add it to the other DNA, or you remove a DNA from one chromosome, one part of it, and add another DNA. So either way, you combine DNA molecules from different sources and you produce a new DNA that is known as the recombinant DNA. So the technology used to do this, or the technology used to produce a recombinant DNA, is known as the recombinant DNA technology. So a recombinant DNA means combination of DNA molecules from different sources. And the technology used to make that recombinant DNA is known as recombinant DNA technology. So if we write the answer here, the technology used to combine DNA molecules from different sources to produce the recombinant DNA is known as recombinant DNA technology. So it is the technology used to combine DNA molecules from different sources to produce the recombinant DNA is known as recombinant DNA technology. Is that clear to you? So then, the next part of the question, write 3 applications of recombinant DNA technology in the field of food and agriculture. So this recombinant DNA technology or genetic engineering is used in many different fields. We want you to write the applications in the field of food and agriculture. What are they? You can produce pest-resistant crops that are resistant to pest attack. Then you can have weedicide-resistant crops. You can produce golden rice. You know what golden rice is? Rice enriched with vitamin A, golden rice. Then you can have tomatoes that are resistant to cold weather. You can increase the yield of crops. You can produce more yield. And also you can have cattle or cattle that can produce more milk, cattle that are more fleshy. Then there can be hens that lay more eggs. Like that, you can have many improved varieties. And also you can have the genetically modified vegetables, fruits with more nutritive values, which are more fleshy, more tastier. That is also possible. Even seedless fruits, those are all possible due to recombinant DNA technology. So you are supposed to write 3 applications. Now we can say pest resistant crops. Then I will say golden rice. That is rice enriched with vitamin A. Then we can have, since it says food and agriculture, cattle that produce more milk. Like this, you can write any 3 applications. You can also say tomato plants that can withstand cold weather. Then weedicide-resistant crops. And also increase the yield of plants. Then the genetically modified vegetables and fruits which are more healthy, which are more nutritive, which are more larger, more fleshy, more tastier, all those can be written here. So I have given you the answers. Pest resistant crops, golden rice, that is rice enriched with vitamin A, and cattle that produce more milk. That is in the field of agriculture and food. I am sure you also have written correct answers. So with that, students, we'll move on to the next question. Extra question 26. The first one, write 3 applications of recombinant DNA technology in the industrial field. So in the industrial field, now say if we need to get amylase enzyme. So the production of enzyme can be done using recombinant DNA technology, where you combine the gene that is responsible for the production of amylase, say into the gene of a microorganism, into the chromosome of a microorganism. So you get a recombinant DNA and then you can produce the enzyme. So production of amylase enzyme. Then another example, production of amino acids that can be used to produce substances like monosodium glutamate, MSG. For that you need the amino acid that is produced using recombinant DNA technology. Then there is the production of vitamins like vitamin B12, vitamin E. Those can be also produced using the technology. Another application is the remedy for pollution. Now there is pollution caused by combustion of fossil fuels, the evolution of different gases into the environment. To rectify that problem, as a remedy to that pollution, you can use recombinant DNA technology. You are familiar with the concept bioremediation, where we use microorganisms that can reduce this pollution. So there we can use recombinant DNA technology. So those are applications of recombinant DNA technology in the industrial field. So if we write 3 applications, one is produce enzymes like amylase. Then produce, I'll say, vitamins like vitamin B12 and vitamin E. So those are examples. Then as remedy to pollution caused by combustion of fossil fuel. So those are all applications. Produce enzymes like amylase, produce vitamins like vitamin B12, vitamin E, then as remedy to pollution caused by combustion of fossil fuel. In addition to that, you can also say produce amino acids for the production of monosodium glutamate. That is also an application. You can write any 3 of these applications. Then we have the next part of the question. Write 3 applications of recombinant DNA technology in the medical field. So here also we can produce different substances. For example, hormones. Now, insulin is a hormone that is used commonly by diabetic patients. Diabetic patients sometimes need to inject insulin as their remedy. So, for that, to produce insulin, we can use the technology. So, then there are other hormones. For example, hormones like growth hormone. So, that also can be produced using recombinant DNA. So, that is again another application. Then if there are genes in the body of a person that has been damaged, that can be replaced. That technique is known as gene therapy. Damaged or disease-causing genes that can be replaced. So that gene therapy, for that also we use the recombinant DNA technology. Then in order to identify samples or match samples to people in the field of forensic science. Now, in the field of forensic science, if there is a murder or a victim found and you need to identify the murderer, then there are samples collected from the scene. In order to match those or identify those, they use this recombinant DNA technology. So, that is in the field of forensic science. So, like that, there are many applications in the field of medicine, medical field. You can write any of those applications. So, here I will say production of insulin, production of growth hormone, and also gene therapy. I am writing short answers because here it is the application of recombinant DNA technology in the medical field. You don't have to explain the applications. If you just write them, that is more than enough. So, application in the industrial field, application in the medical field. Before that, we saw the application of recombinant DNA's technology in the field of agriculture and food. So, you are familiar with all these uses. With that, students, I will move on to the next slide. Extra question 27. A purebred plant produces red-colored flowers while another purebred plant produces white-colored flowers. Red-colored flowers are dominant. Use appropriate symbols to show the occurrence of the second generation in a monohybrid cross of the plants. So, both are purebred plants. So here you can see red colored flowers and white colored flowers, and the red color flowers are dominant. So if you are to write the parents, pure breed, red flower, since that is the dominant color, we will be using capital R, pure breed. Then white flower, since that is the recessive color, we use simple R. So red flowers, white flowers. Then you have to show how the second generation, show the occurrence of the second generation. That is important. So you have to show the F1 generation, and then you need to show the F2 generation as well. So here we will have the parents. Parents both are pure breed parents, capital R, and simple R. Then we have the gametes. Gametes, capital R and simple R. And when they combine, we get the F1 generation. F1 generation, capital R, simple R. So these are all going to be red colored flowers. Red colored flowers. Then they become the parents, or if you carry out a cross between those plants, you will have two plants. Again, that's going to be capital R, simple R into capital R, simple R. So from there we get the gametes. Gametes are going to be of two types. Capital R, simple R, capital R, simple R. And when they combine, you get the F2 generation. Capital R with capital R, capital R with simple R with capital R, simple R with simple R. So capital R, capital R, simple R, again capital R, simple R and simple R. So here this is going to be red, red, red and white. So that is the normal Mendel ratio. Here you have the genotype, capital R, simple R is to simple R, 1:2:1. Phenotype ratio, 3 red is to 1 white, 3:1. So that is the occurrence of the second generation. So here you have pure breed plants. Red color flower is the dominant one, capital R. White flower is the recessive character, simple R. Both are pure breed plants. So those are the parents. Here you get only one type of gamete from each parental plant, capital R and simple R. So therefore the F1 generation gets the heterozygous nature, capital R, simple R, where all the flowers are red in color. Only one phenotype, only one genotype. All are with red color flowers. Then here we have them as the parents, and then you have the gametes, capital R and simple R from both parents, and when that is combined, you get 3 red flowers and 1 white flower. So 3:1 is the phenotype. But the genotype ratio, dominant homozygous, capital R, dominant heterozygous, capital R, simple R, is to recessive, again homozygous, simple R. The ratio is going to be 1:2:1. I'm sure you all also know to write this. So with that, students, we'll move on to the next question. Extra question 28. DNA molecule present in chromosomes are responsible for the transmission of characteristics from generation to generation. That you know, the genetic material or the substance that stores the genetic information is the DNA molecule present in chromosomes. The first question, write the name of the scientist who discovered the structure of DNA. You all know the structure of DNA, the double helix structure, and it was discovered by Watson and Crick. So the name of the scientist, Watson and Crick. Watson and Crick. Then here the second question, what is the structure of DNA? Double helical structure or double helix structure. Double helix structure. Double helix structure. So the scientist who discovered that was Watson and Crick, and it has a double helix structure. I am sure you have written these correct answers. So with that, students, we'll move on to the next question. Extra question 29. Marriages between blood relatives increases the probability of occurrence of sex-linked disorders. Explain this statement. How do you explain that? So hemophilia is a disease where you have the dominant gene. If the dominant gene that is linked to the X chromosome is present, then of course clotting of blood takes place normally. But if that is the recessive gene, double recessive nature, then of course the person has hemophilia, that is where the blood does not clot. But normally in the population, you don't get the double X recessive situation where the female has the disease. But in males, since the combination is X and Y, when the X chromosome has the recessive gene, the male has the disease. So another instance where the female is a carrier and the male has the disease, then also there are more chances that the children will become carriers and also will have the disease. Then the occurrence of children with the recessive gene will be more. So because of that, it is better not to have marriages between blood relatives, as it increases the probability of occurrence of sex-linked disorders. So as an example, consider hemophilia. Philia caused by X-linked recessive gene. So here for hemophilia, if we take the letter H, so what happens? Consider a carrier female and a diseased male. So what happens? Carrier female, X capital H, X simple H. We can either use the lines to show the chromosomes or we can write it as superscript. Either way is fine. Carrier female and the male with the disease. X, Y does not have the chromosome. So what happens to the gametes? Gametes XH and X simple H. Here you have X simple H and Y. And when these combine, when they combine XH, XH, here it is going to be H and Y, and on this side XH, XH and XHNY. Now this is a carrier female. Capital H, simple H, carrier. Then you get X capital H and Y, healthy male, healthy male. And here this is a disease female. But as you all know, like I have said, normally disease females don't exist in the population. When it is the double recessive condition, this fetus gets aborted naturally. So normally we don't get females with the disease in the population. Then we have the last one, again X simple H and Y. So male disease. So now can you see? The mother is a carrier, father has the disease, carrier female, only one carrier male, then disease female, disease male. So out of the 4 children, theoretically the probability of having a healthy child is 1. 1 out of 4 children become healthy. So that means the probability or possibility of getting the disease and the children being carriers is more. 2 children have the disease, 1 female, other one male. But naturally, since the females usually get aborted, there are only males in the population with the disease hemophilia that you are familiar with. So this is how you can explain. Then you have to give a small explanation after that. As shown above, only one child out of 4 is healthy. So it is not suitable to have marriages among blood relatives. So that is how you need to explain. Is that clear? Now when we say blood relatives, that means both the male and the female have that recessive gene. That is why I took the example where the father has the disease and the mother is a carrier. If the father was healthy, then of course the outcome would be different, but still there will be chances that the children get the disease. But the father, if he does not have the gene, the recessive gene, then of course less probability compared to this. But still it is advisable not to have marriages among blood relatives because the probability of getting the sex-linked diseases is higher. So that is how we explain this particular question. So here, marriages between blood relatives increases the probability of occurrence of sex-linked disorders. Explain the statement. Consider hemophilia caused by X-linked recessive gene. Consider a carrier female and a disease male. So both of them have that recessive gene. Then here you can see the carrier female, capital H, simple H in both X chromosomes. Here X is simple H, Y does not have that gene. This is father. Then gametes, you can see XH, X simple H and Y. So when they combine, you get a carrier female, then a disease female who doesn't exist. You have a healthy male and a male with disease. So 1 out of 4 children is healthy. Others either disease or carry. So that is why we need to say, as shown above, only one child out of 4 is healthy. So it is not suitable to have marriages among blood relatives. If you want, you can say, as it increases the probability of getting sex-linked diseases. Is that clear to you all? So with that, students, I'll move on to the next question. Extra question 30. Can the inherited characteristics pass evading one generation to the next generation? Can that happen? Yes. Can you give examples or can you think of examples? Now we saw the Mendel's hybrid cross. When you have the pure breed tall plant, pure breed short plant. F1 generation becomes all tall plants. No short characteristic. Why? Because short characteristic is recessive. But in the next generation, F2 generation, you get the 3:1 ratio where you get tall plants and short plants. So they are the short character skip the F1 generation. So that is possible. Another thing. Now we have discussed about rare inherited characteristics. Now rare inherited characteristics are not common among the population, human population. Why? Because those characters are there, but they skip generations and occur, and also those are not commonly seen. So that is also possible. So the question here is mainly to show that can the inherited characteristics pass evading one generation to the next generation? The answer is yes. Right. Then you are supposed to write the second question. Write 3 inherited characteristics of plants. What are the inherited characteristics of plants? Tall and short character. Round seed, wrinkle seed. White color flower, purple color flower. Or another example, red color flower, white color flower. Color of the pod, yellow color pod, green color pod. Like that, you can write any 3 inherited characteristics. So here we can say height of the plant. Height of plant, tall, short. Then color of flower. Color of flower. Let's say purple, white. Tall, short, purple, white. Then shape of pod. Or shape of, I'll say, shape of seed. Round, wrinkled. Even you can write the shape of the pod. Inflated pod, constricted pod. That is also fine. Color of pod. All those are different characters. Position of the flower. Terminal flower, axial flower. Now, Mendel actually observed 7 characteristics. So, those are characteristics inherited by plants. They are actually, it is the garden pea plant. So, you can write any of those characteristics, or you can write other common characteristics as well. But these are characteristics that you are familiar with already. We have seen how these occur in the first and second generations. So then, the next question. Write the 7 characteristics observed by Mendel in his experiments with the garden pea plant. So again, those are the 7 characteristics that you need to observe. So the characteristics, some of them we have already written. You can write the color of the flower, color of the seed, shape of the seed, color of the pod, shape of the pod, and also you can write the nature of the flower, position of the flower, and also the height of the plant. So I will write all 7 characteristics here. So here we can say color. It can be in any order. Color of flower, color of seed, then shape of seed, shape of pod, the pod, then color of the pod, then you have position of the flower, and the height of the plant. Color of flower, color of seed, color of flower. Flower, color of flower, color of seed, shape of the pod, color of the pod, position of the flower, and height of the plant. So those are the 7 characteristics observed by Gregor Mendel in his monohybrid cross with the garden pea plant. So you should know those characteristics. So with that, students, we'll move on to the next slide. Question number 31, extra question 31. So here, the procedure followed by Mendel for the monohybrid cross of tall and short character was as follows. Fill in the spaces provided. So what was the procedure he followed before we fill in the spaces? Just recall the procedure. He chose a particular character, that is why it's a monohybrid cross, tall and short plants, and they had to be pure breed plants. They were the parents, and there was cross-pollination carried out among them. How did he carry out the cross-pollination? Let's say if it's a tall and short plants, pollens of the tall plants were deposited on the stigma of the short plants. Pollens from the short plants were deposited on the stigma of the tall plants. So only cross-pollination. Then they were allowed to fertilize, and then the seeds were obtained and the seeds were planted. And that was the F1 generation plant. Then those plants were allowed to undergo self-pollination, again production of seeds, and the seeds were planted to obtain the F2 generation. So that is how the cross was carried out. So you know the procedure, now you can fill in the spaces. Cultivation of pure breeding tall and short plants. These dash generation was known as P. What is that? Parental generation. So there you have to write the word parent generation. Parent or parental generation was known as P. You use the letter P. Then blank of tall plants were deposited on the blank of short plants. So what is it? Pollens of tall plants were deposited. Pollens of tall plants were deposited on the stigma of short plants, and pollens of short plants were deposited on the stigma of tall plants. Then, so obtain new seeds by cross-pollination and plant them to obtain the next generation. All plants were what? Tall. Tall, and they are referred to as F1 generation. All are tall plants and they are referred to as F1 generation. Allow to take place within F1 generation. But self-pollination. Here allow self-pollination to take place within F1 generation. The resultant seeds were planted to obtain the F2 generation. the tall to short plant ratio was 3:1. That is how you fill in the spaces. So here if we quickly read the procedure, cultivation of pure breeding tall and short plants, these parent generation was known as P. Pollens of tall plants were deposited on the stigma of the short plants and pollens of short plants were deposited on the stigma of tall plants. Obtain new seeds by cross-pollination and plant them to obtain the next generation. All plants were tall and they are referred to as F1 generation. Allow self-pollination to take place within F1 generation. The resultant seeds were planted to obtain the F2 generation. The tall to short plant ratio was 3:1. So that is the Gregor Mendel's monohybrid cross of tall and short plants, pure breed plants. So with that, students, we'll move on to the next slide. Extra question 32. The monohybrid cross of pea plant considering tall and short character can be expressed using symbols as below. Write the appropriate terms in the spaces below. So here the diagram is given. You can see the parents, pure breed tall plants, capital T, and pure breed short plants, simple T, simple t. Then what are these? They are the gametes. Then you have the first generation, capital T, simple T, all are tall plants. So when there is self-pollination among them, again you get the gametes, and finally you have the F2 generation, capital T, capital T, simple T, simple T, and simple T. Those are the F2 generation. 3 tall plants and 1 short plant. So here you need to write the names corresponding to different stages. So here, what is this? The parent generation. So we are supposed to write appropriate terms in the spaces below. Parents. Parents is going to be 1. Then what is 2? This is going to be 2. We have the gametes. And here, this is 3. That is going to be the F1 generation. And then here we again have 4. That is gametes. And the last line, 5. That is going to be the F2 generation. That is how we have the different terms. Then, the next question, what is meant by a monohybrid cross? What is a monohybrid cross? Cross between organisms considering only one character at a time. For example, height of plant is a character. It can be tall or short. If you consider only that, that is a monohybrid cross. So what is meant by a monohybrid cross? So here we can say cross breeding plants considering a single pair of contrasting characters. So that is a monohybrid cross. So monohybrid, a single pair, that is the important thing. So here crossbreeding, you can even leave this term plants because it's common to all organisms, but here we have done all these experiments, or Gregor Mendel has done all experiments using the garden pea plant. So it is a monohybrid cross, crossbreeding considering a single pair of contrasting characters. Tall and short, round and wrinkled, white and purple, so contrasting characters. So that is a monohybrid cross. I'm sure you all can understand that. So with that, students, we'll move on to the next slide. Extra question 33. Round seed is the dominant character in the monohybrid cross of pea plant. Considering round and wrinkle seeds, show the occurrence of F2 generation using the Punnett square. So you can use symbols to show, or you can use the Punnett square. So there also you need to have the parents, then gametes, but to indicate them we use squares. We can use a square to indicate the parents. So here the characters are round seed and wrinkle seed. So capital R dominant character, simple R recessive character. So from here we get the gametes. Gametes, you will have capital R and simple R there. They are the gametes. Then you have the F1 generation. F1 generation, again you need to use a square, but only thing is you just divide them like that, because capital R, simple R, to only one type of gamete from each parent, and combination is this, all round seeds. All round seeds. Then after that, you get the gametes from this generation, and then you get them to undergo self-pollination, and you get the F2 generation. There, of course, for the F2 generation, again you draw a Punnett square, but you need to have the gametes. So 3 columns and 3 rows. So capital R, simple R, capital R, simple R. All are heterozygous. Then the combination of R and R, capital R, capital R, capital R and simple R. Then you have the simple R here, capital R there, but we write the capital R first, then simple R and simple R here. So you get the F2 generation, homozygous dominant, heterozygous dominant, and heterozygous recessive. That is how you show using the Punnett square. Is that okay, students? So we have the parents, squares, capital R, simple R, simple R. Gametes usually in circles. So then when they combine, you get the F1 generation. R and R combine to give heterozygous dominant character round seeds. And then from there you get the F2 generation. 2 types of gametes, capital R and simple R in both parents. So you get this type of generation, occurrence of the F2 generation. I'm sure you all also have written the same answer. So with that, students, we'll move on to the next question. Extra question 34. Given below are the result of an experiment carried out by Mendel in hybridizing pea plants. So here you have the character is the color of the pod, then hybrid is between green and yellow, and F1 generation is all green. That means green is the dominant character. And F2 generation you have green 428 plants and yellow 152 plants. So the questions related to that. Write the genotypes of the pure breed parents, green and yellow. So you know green is dominant. So green pod has to be capital G, capital G. then yellow pod should be simple G, simple G. They have to be homozygous and you have to show the dominant and recessive characters. Then identify the dominant and recessive characters in the above. Green pod, pod dominant, yellow pod recessive. How did we identify that? From the F1 generation. In a Mendel's monohybrid cross of purebred plants, always the first generation, F1 generation, consists of the dominant character. So, green is the dominant character. Then yellow pod is the recessive character. And also F2 generation, you can see the ratio. Green is to yellow has to be 3:1. 428 is to 152. So, you can see green is higher in number. Then write the phenotype ratio of F2 generation. Phenotype ratio. What is a phenotype ratio? It is the character that is expressed in the generation, or the external characteristic of an organism is what we call as phenotype ratio. So, your phenotype ratio green is to yellow, you know it is 3:1. Even if you divide 428 by 152, or if you just multiply 152 by 3, now if we try that, 152 multiplied by 3, because this is 1, so 3 times have to be green. 6, 15, 3 plus 456. So here you can see they have obtained 428, and you all know it is the closest ratio between the plants. 428 you should have, if it was exact, it should have been 458, but they have obtained 428 plants with green color seed. So there the ratio will be 3:1. So even if you find the ratio using the values there, you will still be writing the ratio as 3:1. That is the closest whole number ratio. Is that clear to you? That is how the monohybrid cross of purebred plants occur. So I will move on to the next slide. Given below are the result of an experiment carried out by Mendel in hybridizing pea plants. So this is the same table that we had before, and this is the fourth part of the question. Use a Punnett square to illustrate the occurrence of F2 generation from F1 generation. Write the phenotype and genotype ratio of F2 generation. So here you only need to use one Punnett square to show the occurrence of F2 generation. So there F1, you can remember. Now from F1 to F2 you have to have a square like this, and in this F1 all are green plants, but you all know they are heterozygous. Capital G, simple G. So from there you get two types of gametes, capital G and simple G, capital G and simple G. When they combine you get capital G, capital G, capital G, simple G, again capital G, simple G, simple G, simple G. So what is the phenotype ratio? Phenotype, the external feature, green is to yellow. Green is to yellow, 3:1. Then you have the genotype ratio. Genotype ratio, the combination of genes. Capital G is to capital G, simple G, is to simple G, simple G. You have 1, you have 2 of these and 1. So that's going to be 1:2:1. So you're familiar with the Mendel's ratio. But you can again, when you write also, you can always check and confirm your answer. So phenotype ratio is 3:1. Genotype ratio is 1:2:1. Is that clear, student? So then we will move on to the next question. Extra question 35. A diagram relevant to the experiment carried out by Mendel on inheritance using garden peas plant is given below. Fill in the spaces. So the parents, phenotype, round seed, wrinkle seed, same as before. Genotype, they want you to write the genotype. The spaces are given. So you know round seed. Now here again, F1 generation, all round seed. So that means round seed is dominant. Anyway, Mendel's cross, you know the characters and you know what is dominant and recessive. So here capital R, capital R. Wrinkle seeds have to be simple R, simple R. And from the gametes also you can identify. Capital R, simple R. So this has to be homozygous capital R and that has to be homozygous recessive character. So gametes are given. Then F1 generation, phenotype all round seeds and genotype it has to be heterozygous, capital R, simple R. So we have completed the diagram or how the first generation occurs from the parents. So with that, students, I have come to the end of this chapter and also the end of this lesson. So with that, we have completed the lesson Inheritance.
To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.